Viruses are a unique group of biological entities known to infect every type of cell. As they are extremely abundant on our planet, one can simply ask, how can a very small particle be capable of causing disease and death? Their structure, and should we say unusual or unpredictable behavior, have made a big impact to the rest of the microbial world. As much as we consider viruses as poisons, their influence on the genetic makeup of bacteria, plants and animals led to the evolution of many life forms. Our class objectives will lead us to understand the nature of viruses, identify selected important groups of viruses in associated diseases, and lastly discuss cultivation of viruses. Viruses are considered as cellular microorganisms together with prion and viroids. They do not exhibit most life processes of cells, and because complete virus particles called virions are very small and simple in structure, scientists were unable to see viruses until electrical microscopes were invented in the 1930s. Most viruses range in size from 10 to 300 nanometers in diameter. Viruses are not alive, but they are the major players in the ecosystem. No type of organism is safe from viral infection. That's why there are animal viruses, plant viruses, mycoviruses, and even bacteriophages. Viruses are said to have 5 specific properties that distinguish them from living cells. Number 1. The vast majority of viruses possess either DNA or RNA. Unlike living cells, which possess both, they are unable to replicate on their own. Their replication is directed by the viral nucleic acid once it has been introduced into host cell, and unlike cells, they do not divide by binary fission, mitosis or meiosis. They lack the genes and enzymes necessary for energy production, and lastly, they depend on the ribosomes, enzymes, and metabolites of the host cell for protein and nucleic acid production. A typical variant consists of a genome of either DNA or RNA surrounded by a protein code called capsid, which is composed of many small protein units called capsomeres. Together, the nucleic acid and the capsid are referred to as a nucleocapsid. Some viruses, called enveloped viruses, have an outer envelope composed of lipids and polysaccharides. These protruding molecules, called spikes when they are on enveloped viruses, are essential for the attachment of viruses to the next host cell. Viruses are classified by the following characteristics. Type of genetic material, either DNA or RNA. The shape of the capsid, number of capsomeres. The size of the capsid. Presence or absence of an envelope. The type of host cell that it infects, the type of disease it produces, its target cell, and its immunologic or antigenic properties. This table shows the most common viruses, their nucleic acid type, shape, and sizes. Capsids of viruses have various shapes and symmetry. They could be helical or coiled tubes. They could be bullet-shaped, spherical, or a complex combination of the shapes. In some polyhedral capsids have 20 sides or facets. Geometrically, they are referred to as icosahedrons. Here's a table of some of the selected important groups of viruses, their characteristics, and diseases they cause. The genome of most viruses is either a double-stranded DNA or a single-stranded RNA, but a few viruses possess both single-stranded DNA or double-stranded RNA. One example from this table are the pox viruses, which are large brick-shaped enveloped viruses. It has a double-stranded DNA variola, which causes smallpox, and vaccinia, which causes cowpox. Are some of the viruses under this type. Take note of each of their morphology and the diseases associated with the viruses. As you have observed, capsids of viruses have various shapes and symmetry. This is another illustration of the morphological characteristics of common viruses. You have learned from your previous lectures the modes of viral multiplication. The process starts from the transmission of the virus, the invasion of the virus to its host, up to the responses of the immune defenses and human attempts to control viral infections. The host cells are destroyed as the virus multiply within the infected person. This cell destruction leads to most of the symptoms of the viral infection, which vary depending on the location of the infection. Thus, viruses are specific as to the type of cells that they can infect. For these reasons, certain viruses cause only respiratory infections, whereas others cause only gastrointestinal infections, and so on. One of the primary reasons why viruses undergo cultivation is for the development of vaccines. However, before all the systems of cultivation have emerged, scientists were unable to grow specific viruses because of its need for a natural host. Before we proceed, watch the short video clip about how a British doctor helped eradicate a deadly disease by introducing vaccination to the world.
10,000 years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her odd treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected, but it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairy maids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family. But when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the dairymaid Sarah Nelms, Blossom the cow, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
Aside from some gram-negative bacteria, viruses are also obligate intracellular pathogens. This means that they can survive and multiply only within living cells called host cells. It was a challenge for microbiologists to grow these pathogens because they are unable to thrive on artificial or synthetic media. To grow such organisms in the laboratory, they must be inoculated into embryonated chicken eggs, laboratory animals, or cell cultures. Just like bacteria, viruses need to undergo cultivation to further isolate and identify viruses in clinical specimens and do detailed research on viral structure, multiplication cycles, genetics, and defects on host cells. But most importantly, cultivation prepares viruses for vaccines. Vaccines have significantly reduced the prevalence and impact of many infectious diseases that were once common and often deadly. Viruses can be cultivated in a variety of ways. First, it can be cultivated using live animal inoculation. The animal is exposed to the virus by injection of a viral preparation or specimen into the brain, blood, muscle, body cavity, skin, or foot pads. Specially bred strains of white mice, rats, hamsters, guinea pigs, and rabbits are the usual choices for animal cultivation of viruses. Another way is by using bird embryos. It furnishes several embryonic tissues that readily support viral multiplication. It is an intact and self-supporting unit compete with its own sterile environment and nourishment. Chicken, duck, and turkey eggs are the most common choices for inoculation. Lastly, using cell or tissue culture. Animal cell cultures are grown in sterile dishes or bottles with special media that contain the correct nutrients required by animal cells to survive. The cultured cells grow in the form of a monolayer that supports viral multiplication and permits close inspection of the culture for signs of infection. Again, in live animal inoculation, the animal is exposed to the virus by injection of a viral preparation or specimen into the brain, blood, muscle, body cavity, skin, or foot pads. While when using bird embryos, the egg must be injected through the shell, and the exact part of the egg that is inoculated is guided by the type of virus being cultivated and the ghost of the experiment. For example, amniotic inoculation is used for influenza and mumps virus, while yolk SAC inoculation is used for the herpes simplex virus. Another way to cultivate virus is through the use of cell cultures. Cell cultures can be classified as primary cultures or continuous cell lines. Primary cell cultures are prepared by placing freshly isolated animal tissue in a growth medium. Embryonic, fetal, adult, and even cancerous tissues have served as sources of primary cultures. One disadvantage is that they can generally have a limited existence and eventually they will either die out or imitate. Another type of cell culture is the continuous cell lines. They tend to have outer chromosome numbers, grow rapidly and show changes in morphology. They can be continuously subcultured, provided they are routinely transferred to a fresh nutrient medium. The newest type of cell culture has helped in the faster development of influenza vaccines, which has been grown in chicken eggs since the 1950s. Around 2009, scientists succeeded in propagating the viruses in a continuous cell line derived from animal kidney cells. Since then, influenza vaccines have been produced from both cell culture and from chicken eggs.